Good afternoon, everyone. I am Gerald van Eeden, and I would like to give you an overview of the findings for my master's project, followed by what I'm planning to do for my PhD project. And both follow the same theme, which is on the slide, expanding existing tools to aid in the epidemiological study of populations with complex ancestry. I've had a lot of practice with that word. So a recombination map is a genetic map that illustrates the variation of the recombination rate across a region of the genome or the entire genome. Thus, it has information regarding the physical distance and the genetic distance of a genetic marker. Previous analysis on recombination in mammals have shown that crossover events tend to cluster together in areas known as hotspots. In these areas, the recombination frequency tends to be many folds higher than the surrounding genome. In the figure, the recombination rate is plotted over the physical position of chromosome 11. Here, the peaks illustrate hotspots. Some studies indicate that 80% of all recombination events could be found within these hotspots. Currently, there is a lack of high-resolution population-specific recombination maps for Southern African populations. I set out to compare various methods to infer population-specific recombination maps during the first year of my master's. There are currently 4 methods used to infer recombination rates: pedigree-based methods, LD-based methods, methods that use LD summary statistics, and methods that rely on identity by descent. I tested the first 3 methods, and the IBD-based method was only developed within the last year, so I did not test it. However, it appears to be very accurate at high resolutions and with a low number of individuals. For my comparison, I settled on using FastEPRR, an LD summary-based statistics method to infer population-specific recombination maps, because I don't have 100,000 individuals like the pedigree software requires, and it produced comparable results to LD-based methods at relatively high resolutions. So I compared a recombination map for the Nama, which is a Khoisan population, to recombination maps of various populations from different continental groups. And here are the results of that comparison. The figure shows the correlation of the recombination rate between various populations at a 50 kilobase resolution in the upper triangle and a 5 megabase resolution in the lower triangle. I found that the relative correlations between populations are related to the continental levels of population differentiation. Interestingly, the Nama recombination map was the most distinct at a 5 megabase resolution. I also did a preliminary analysis of the effect of recombination map choice on local ancestry inference. The figure shows the ancestry switches of a Nama individual as you traverse chromosome 5. The blue line was inferred using a recombination map inferred for the Nama, and the pink line was inferred using the combined HapMap2 recombination map. This map is often used for local ancestry inference in the Nama. It seems like the recombination map choice affects the placement of ancestry switch points, and this could affect downstream analysis, since local ancestry calls are often used to compensate for ancestry in association studies. For my PhD project, I aim to do the following. I would like to determine the optimal recombination map to use in local ancestry inference for a given population. I'd like to characterize the appropriate use of ancestry information in genome-wide association studies. And finally, I'd like to determine the most prevalent PRDM9 variant in the Nama. Given the time, I'd like to focus on my initial results for AIM3. But first, PRDM9 is a meiosis-specific methyltransferase with a tandem repeat zinc finger domain. PRDM9 determines the placement of recombination events in some mammals, including primates and mice. The repeat region of PRDM9 is what determines the particular binding motif. To date, more than 50 PRDM9 variants have been found, with the most common variants being A, B, and C. Very convenient. A and B share binding motif, but have different binding affinities. It has been found that approximately 40% of hotspots contain the binding motif highlighted in red. We conducted variant annotation of 84 self-identified Nama individuals, and found that 73 of these individuals contain the PRDM9B variant. 30 of those individuals are heterozygous for a non-synonymous G2C mutation, and 26 out of 30 have local Khoisan ancestry. This change is predicted to affect PRDM9's binding affinity, which could lead to lower overall recombination or less motif-specific recombination. We also inferred hotspots genome-wide for 54 unrelated NAMA and 3 other populations. A hotspot is defined as a 1 to 2 kilobase region with a several-fold higher recombination rate than the surrounding genome. The threshold used in the results in this table is a 5-fold threshold. Interestingly, the NAMA has surprisingly fewer hotspots. So this could be due to various reasons. One, and I think this is probably it, is I did something wrong. Then there's the possibility that PODM9 is responsible. And then finally, the NAMA's relatively large LD blocks due to recent admixture are actually what's affecting the inference of the software. And that is it. I'd like to acknowledge the following people as well as welcome any of your questions.
Thank you very much, Gerald. This it looks like a really exciting project. I think it's something that does need to be tackled in South Africa and Africa in general. I'd like to know if you could just elaborate very briefly on how you're going to do your second aim. I think that involved GWAS, and if I remember correctly.
Sure. So for my second aim, I would just like to, there have been many of these reviews in the past, but I'd like to do another systematic review of how exactly people use or researchers use ancestry information to correct their GWAS. But I would like to focus on local ancestry inference specifically as a method to do this. And there haven't been a lot of studies in that area. So the focus of that aim would be developing a protocol for how local ancestry inference can be incorporated into GWAS.
Okay, thank you. A question from George. He says, great talk, wow, and something we need for our African work for sure. He says, I know African LD blocks tend to be shorter than other populations. Do you need to adjust or train the tools to expect this?
Well, that would depend on the question. So given hotspot inference, that would probably be the case. You'll probably find less intense hotspots in populations with higher LD. So yes, in some instances you would need to adjust for it. However, when running the software itself, you don't need to adjust for it. It's more like a post-analysis step.
Okay, great. Thank you so much.